Honored guests, esteemed colleagues, and dear students. Welcome to the distinguished lecture series organized as the part of the 70th anniversary celebration of Middle East Technical University. As a leading research university, METU has long been committed to advancing scientific excellence and fostering meaningful dialogue between science and society. And it is a great pleasure to have you all with us today for this special event. We are honored to host two distinguished guests today. The 2022 Nobel laureate in chemistry, Professor Morten Meldal, and Dr. Phaedria Marie St. Hilaire, an accomplished leader in STEM with extensive experience in pharmaceutical and biotechnology sectors. We look forward to an inspiring and engaging program through the day. To begin our program, I would like to invite Mr. Erhan Atıcı from the Turkish Chemists Society to deliver his introductory remarks.
Dear President, dear colleagues, dear scientists, dear professors, dear students, and our honored guests, Meldal and Phaedria. Firstly, welcome. It's a great honor and a great pleasure for us today to see you here. We are here gathered under the power of science. I want to extend my sincere regards to you, to each of you on this hall. Since its establishment, our Turkish Chemists Society, we always support the mission of the contributing to academic field and the industry of chemistry. So we perform many organizations during the years. We do 7 major congresses in a year. It is drug chemistry, food chemistry, environmental chemistry, so on. And also for the first day of the students who enter their professional life, we perform white coat ceremonies. And also we do career talkings for the last year students to guide them, to encourage them for their professional lives. So I think we do very solid phase for the science to gather together all these experiences, and we are serving for the chemical industry. Science is a journey, and begins with curiosity, and grows with patience. Patience supports the growing, and then it gains remain meaningful when you serve the humanity with your science, with your educations. Especially chemistry, in particular, it is a main transforming pillars for that area, for that journey. Because chemistry, with its presence, it touches every point of life. Wherever you see the chemistry. From health to environment, from defense to advanced technologies, chemistry is everywhere. Therefore, every contribution to the chemistry is investment to the future of humanity. We should be. So we are aware of this, as our society, we are aware of this one. So with this responsibility, we not only follow the science, we also producing, sharing, and developing. Supporting our young chemists, supporting young colleagues, encouraging them, guiding them, preparing them for their great goals. So it is a commitment deeply that we believe in. Today, we have two very great honors here, very exceptional scientists. Nobel laureate, Professor Morten Meldal, together with us, and Dr. Phaedria, together with us. Actually, his schedule is very crowded. He gave me appointment for 20 months later for being here. He came to here. I want to thank to him. He will share his knowledge, his experiences, his pioneering works, and we will listen. We will learn many things from them. So there is an opportunity to benefit from him, from his knowledge, from experiences. It's a very important for us for the students, for everybody here. So we are here, the power of the science, I think, again. Dear colleagues, we firmly believe that if there is the science, there is the future. If there is the chemistry, there is the transformation. So because you are here, we have hope. You know everything in the world, many things happening in very short times. So we have hope, I think, because there is science. So to sum up, I want to say my sincere regards to Meldal and Phaedria welcoming here, and very thanks to University, Middle East Technical University. They host these events, and thanks to everybody who take part in this event, and thanks to everybody, and thanks listening to me. Thanks for welcoming. Thank you.
Thank you, Mr. Atıcı, for your valuable remarks and for your continued support. Now it's my pleasure to invite METU Vice President, Prof.Dr. Tülin Gençöz, to deliver her welcome speech.
Thank you. Distinguished guests, esteemed colleagues, dear students. It's my great pleasure to welcome you all to this special session of the Distinguished Lecture Series organized as part of 70th anniversary celebrations of Middle East Technical University. Welcome all. Today is truly an exceptional occasion for our university. We are deeply honored to have two distinguished guests whose accomplishments and leadership have made a profound impact in their respective fields. It's a privilege to welcome Professor Morten Meldal, recipient of 2022 Nobel Prize in Chemistry, and Dr. Phaedria Marie St. Hilaire, an accomplished leader in the pharmaceutical and biotechnology sectors, as well as a globally recognized advocate for diversity, equity, inclusion, and belonging. Professor Meldal's groundbreaking work in click chemistry has revolutionized the way chemists think about molecular design and synthesis. His contributions have not only advanced fundamental science, but also opened new pathways to innovation in fields ranging from drug discovery to material engineering. His achievements exemplify the highest ideals of scientific excellence and curiosity. Professor Meldal, we are truly honored to have you with us today. Your presence, if you like, your presence is an inspiration to our entire academic community, and especially to our students and young researchers who aspire to contribute to science at the highest level. We are equally delighted to welcome Dr. Phaedria Marie St. Hilaire, with her remarkable career spanning global leadership roles in the pharmaceutical and biotechnology industries, and her impactful work in advancing inclusive and equitable organizational cultures. She brings a unique and highly valuable perspective. We look forward with great interest to her lecture this afternoon, where she will share her insights and experiences with us. As Middle East Technical University, we take great pride in bringing together leading scientists and thought leaders with our community, fostering dialogue, inspiration, and collaboration. Before I conclude, I would like to extend our sincere gratitude to the Chemists Association for their valuable contribution. I would also like to extend special thanks to our esteemed alumnus, Mr. Erhan Atıcı, for his support and commitment to this event. We are truly privileged to host such distinguished speakers, and we very much look forward to Professor Meldal's lecture this morning, as well as Dr. St. Hilaire's talk later today. Finally, please allow me to convey our rector's warm greeting. Unfortunately, he is unable to join us at this moment due to an official meeting. He has asked me to extend his sincere regards to distinguished guests and to all participants, and to express his deep appreciation for this valuable gathering. I know you had a chance to meet him this morning, in the morning, and he also looks forward to meeting our esteemed speakers later this afternoon. Thank you very much, and I wish you all an inspiring and engaging session. Thanks a lot.
Thank you, our professor, for your kind words. Now we will proceed with a brief introduction of our distinguished speaker, Professor Morten Meldal.
Welcome to the Nobel Talks, proudly presented by Turkish Chemists Society and Middle East Technical University. A mind that reconstructed our world at the molecular level, redefining the very boundaries of science. 2022 Nobel Laureate in Chemistry, Professor Morten Meldal. His journey did not begin in state-of-the-art facilities, but rather with a pure, unfiltered curiosity about the natural world on a small island.
I was very, very lucky because my grandparents have a farm in Bornholm where I was shipped off to every single opportunity they had. So I was there, and I was out in the morning at 8 and in at 9 o'clock in the evening. So I had a whole day of nature and, being in the fields, in the forest, in the beaches and so on. It was really, really nice as a child.
That drive to understand nature's kinetics soon evolved into a passionate and sometimes explosive interest in chemical reactions.
Later on, when I was 16, I ventured out to a small company that sold all sorts of chemicals and bought what you would consider today to be explosives. But eventually I made something that exploded in the backyard of my parents' house, and they actually sort of stopped the exercise at that point. But it created a lot of interest in me for understanding the chemistry, how it works, the reactions, the kinetics, and why is it that chemistry actually happens.
That teenage curiosity eventually triggered a revolution in modern science. Professor Meldal is a pioneer of click chemistry. Traditional chemical synthesis is often slow, complex, and generates unwanted byproducts. Professor Meldal engineered a flawless alternative. By introducing copper as a catalyst, he discovered a method to bind two specific molecular groups, azides and alkynes, with absolute precision and instantaneous stability. This reaction is exceptionally highly yielding and specific, resulting in zero chemical waste. Today, click chemistry serves as the architectural foundation for groundbreaking advancements. It enables targeted cancer therapies, precise DNA mapping, and the synthesis of advanced industrial materials. It is the epitome of pure, efficient science. For rendering complex chemical reactions entirely controllable and pure, he was awarded the highest honor in science, the 2022 Nobel Prize in Chemistry.
When I received the message from Stockholm on the Nobel Prize, that was really cool. First I thought it's probably a prank played by my students, because I've tried that before. But I could very, very quickly hear that this was not a prank and this was serious. And I talked with 3 committee members, and they told me about it, and it was very, very exciting. Then I had to wait for an hour, and that was kind of tough, to wait for an hour not telling anyone. Even my wife I couldn't call. And after that I heard the announcement, and as soon as I opened my office door, there was like hundreds of people in the corridor outside, 50 people in the corridor outside, all cheering, and that was very, very pleasant experience.
Despite global recognition, Professor Meldal's true driving force remains far from the spotlight. He is an academic at heart, deeply committed to the laboratory and the next generation of scientists.
I actually work still in the laboratory, and I work in the laboratory because I think it's fun. I'm not driven by ambition, I'm not driven by another Nobel Prize. I'm driven because I like to investigate chemistry. I like the understanding of the world that we are living in.
a scientist who proves that true innovation comes from stepping into the unknown. We are profoundly honored to present the Nobel Talks, bringing world-changing ideas to the next generation of researchers, because the pursuit of knowledge never truly ends. Welcome to the Nobel Talks.
Ladies and gentlemen, please join me in welcoming the 2022 Nobel laureate in chemistry, Professor Morten Meldal, to the stage.
Hello, ODTÜ. So thank you to Erhan and to Tulin for this kind introduction. It's a pleasure, and now I just have to explain some things. My lecture's already been given, so now I just have to explain a few things. And the first thing I want to say is that chemistry is everything. That is not only a language that we use to describe the world, it's actually everything. So chemistry is the molecules all around us that interact, and every single thing that happens, every process that occurs is chemistry at work. So when we feel hungry, it's chemistry. When we feel in love, it's chemistry. When we are tired, it's chemistry. All of that comes from molecular interactions. Our materials around us is chemistry. We are changing the world at an unprecedented pace at the moment, and this happens with chemistry. We are using chemistry to change the surface of this world. So chemistry is ultimately very, very important for us, not only because it is the world around us, but also because it is an existential philosophy upon which we can actually rely for reality, to understand it and to be better in utilizing this understanding in our world. So that is why I have the noble quest of realizing potential chemistry in a global society. And I received the Nobel Prize in 2022, as you heard. It was sort of a surprise. Actually, I had a phone call the week before from a company in Sweden that wanted to sell me instruments, and they had been very persistent. So I was kind of a little bit irritated. So when I saw the Swedish number, I go, yes! And there was a long pause in the other end of the phone. And then I say, this is the Swedish Nobel Committee. So I could hear this was really Swedish, and I understood that this was actually the Nobel Prize. Great surprise. I would like to say it's such an honor and a privilege for me, old as I am, 72 years old, to be able to speak to all of you young people. Because for me, the most important thing is that we help the young people understand this chemistry universe. So that is why I think that not only is chemistry everything, but we need to transform our society so that the youth actually learn this from the very first grades in school. If chemistry is everything, it's more important even than writing and reading and math. So we need to teach them this, and we can do that because the children have this amazing ability of image storing. So the children learn from when they are born, they learn by observing the surroundings. This very complex three-dimensional world around them is stored in their brain. They make a mirror image of that, and that's how they can navigate this world. We can do the same with chemistry today because we can animate chemistry. We can make them understand chemistry as a three-dimensional world where they instinctively know exactly what is happening in the chemistry world. Meaning that they would understand what global warming means in reality, not by the fantasy of some people around the world, but in reality, what does this mean to us and our future? Also, all big discoveries, in my opinion, are serendipitous. If a discovery is serendipitous, that means that we cannot really plan it. We cannot sit down and write on our office desk, we want to do this for 5 years, send in the application, and then do that for the next 5 years. Because this discovery is not coming like that, it's coming by observation in the laboratory. Something new like penicillin, click chemistry, other things. All serendipitous because it's an observation of something that is not expected. If it was expected, it's probably done a long time ago. So that is also why we need funding by performance, a different type of funding system. Not these office applications that goes in and we get it funded, but actually funding what happened the last 3 years was very good, so let's fund the next 3 years. Life is composed of only 16 atoms, so this chemistry that we are talking about that makes up us, that chemistry is made from only 16 atoms. And the thing is, these 16 atoms, they contain the ability to form life, and they will do so whenever there is the conditions that is needed to make the atoms, form the bonds, make them make molecules, make these molecules go together, make complexes, nanostructures, cells, life. That's what happens here. So it's just protons and neutrons surrounded by electron clouds, and they shared between the nuclei and molecules are formed. It's only carbon that we can imagine can make life, and that's because carbon is in the center and it's neutral, can form bond to itself, very strong bonds, and also can make double bonds, which silicon, for example, cannot do. So they make these nanostructures, and the electronic waves of the molecules, they actually respond to the environment. We know that from NMR. If we put into one solvent, we see one spectrum, another solvent, we see a different spectrum. That's because the solvent interacts with the molecules, the electrons in the molecules, and you get different spectra. That means that every single molecule, every single carbon atom in this room is a little bit different because the environment is different. That's what makes up the possibility of make complex life. So we are working in our lab with molecular recognition, with molecular transformation, signaling, transport, but we don't work with DNA and RNA. But the other 4 very important things for life we are working with on all the time. So as I said, my inspiration came from Bornholm, or was in the video, and this is a forest from my grandfather. And you can see here, all these trees know where each other are. The insects know where to go, and so on. That's very exciting, and how do they do that? That was really my first interest in nature, not chemistry, but nature, was how is this happening? Why is this so beautiful? What is it that makes this synergy? And the answer, of course, is the chemistry. So I got into that later on, and as was said, I went to this shop and bought the gunpowder, big box like that, and went home in the train with 15 kilos of explosives. That would not be possible today. And back in the basement, and we made this kind of things, very, very beautiful rockets, actually. I have a whole lab book this thick with prescriptions to do beautiful things. Well, for me, factual knowledge about chemistry, knowledge about physics is very important, but it's not the most important thing. The most important thing for me is the soft skills. Soft skills in the people that I have to hire has to include intuition, imagination, association, ability to observe. Very, very important, because if you are preoccupied with what you expect, you're not seeing the unobvious. And then, of course, you should have creativity so you can combine stuff. Most importantly, you should have courage, because courage allows you to tell your PI that he is completely wrong with his office desk suggestions, and you can see something else in the lab. And the road to scientific success is never straight, so you have to persevere. Do it again and again, and eventually you will succeed. You make small adjustments along the way, and you get to the success. And you have to be able to step out of your comfort zone here. The comfort zone, you have to step out of it, do something that is completely foreign to you, but can be added to your already existing knowledge, and you expand, you study all your life, and you get more and more clever, taking your experience from one field to the next. So this here is T-Rex. We can call him Donald T-Rex. And Donald is, as you can see, not a very empathic creature. Donald also has a very small brain compared to the size. And Donald is controlled mainly by aggressor molecule like adrenaline and noradrenaline that controls the blood flow in his huge body. And then he has also the dopamine that comes when he has success in capturing a prey and eating it. And serotonin is very important, maybe not for Donald, because this is a cognition molecule that allows us to operate in an optimal way in our environment. So people in general are hurt people. So they are hurt communities of individuals that lives from their success is a result of their empathy and their, they like each other and they help each other. That is the key to our success. And this is a love molecule, oxytocin. Both men and women have it, women more than men. This is what the structure looked like in 3 dimensions, but it actually doesn't look like that, and that's where the children should learn that it actually looks like this, a piece of chewing gum with colors, and that is the electron densities that you see in red and blue. More is red, less is blue, and this is how it then interacts with the opposites in a receptor, for example. So here we have the binding of oxytocin to the receptor, sends a signal in here, and we get empathy, love, pleasure, nesting, and all of that. Very important interaction. And again, you can see all these emotions that you are having, they're actually controlled by chemistry. And then nature is quite cheap, so here you have a natural product that looks very much like this one, only 2 small changes, and now we have a molecule that controls heart pressure. So maybe there's something about love and hearts, I don't know. Well, we thought this is a very exciting field. So I would like to work inside this field with peptide materials, drugs, nanostructures, and so on. All of these fields where peptides play a pivotal role because peptides is the main controlling tool in nature. And we would like to do this in combination with organic chemistry. So we called it quantitative chemical transformations, which is a long and boring sentence. Instead, click chemistry is a much better one. But we combined this quantitative chemical transformation with peptide diversity and made a lot of different new molecules that could add to our stem. So we built this technology around our polymers. These are hydrophilic polymers that swell up in water. We can also do organic chemistry on them. So this is for solid phase synthesis. And we transformed all of these organic chemistry reactions to be quantitative on solid support together with the peptide chemistry. And we could investigate on these beads the reactivity of all of these enzymes. So that was very cool, that we could combine the chemistry, the enzymology, and everything on one bead. So each of these small beads becomes a reactor, a monitoring device. So this is important. And you will notice here we have the QAG reaction, 1 out of 30 reactions that worked extremely well. and led to the Nobel Prize. So we built this platform where we're doing combinatorial chemistry in these reactors. We can make 500,000 molecules in a few days. And then we test the molecules like this, find the active ones, and find out what they did with the enzymes. We can resynthesize, these are now quenched substrates, and we can synthesize those, inject them into a mouse, and see when the enzyme that we tested here, when that is active in an arthritis model. We can also grow cells on the surface of our beads. So this is like now we have the monolayer of cells on the surface that acts as a detector for cell-compound interaction. We can see one here that is active on the background of inactives, and we can take that out, isolate the molecule and identify it, and that is a very tedious process. So we also developed encoding where we put small microparticles inside the bead. And we can read this matrix of microparticles just like it is a barcode. And we can do that every time we do a reaction or when we do an assay at the end. So we use this technology to build peptides like this one, very difficult ones, where we were only able to do that with using acetoacid chlorides, so amino acids with an A site as a protecting group, and we built these molecules. We thought that was cool because we could not build them in any other way. But I also thought maybe we can use that to introduce a triple bond in the center of a long peptide hormone, for example, and then click on side chains like you have it in Wegovy and those new peptide hormones that are really doing successfully now. But we were not able to actually do that. We could not introduce this into the peptide by this method. So we make the copper acetylide, and then when we react it with that one, we only get a single product, and it wasn't this one. So Christian Thorne, who was my PhD student at the time, he came to my lab and said, "This is really an office program. I don't want to do it anymore. I want to go to Novo Nordisk and work with real chemistry." So I said, let's look at it. And I found this proton in his spectra, and we decided, well, this is what is formed even in the presence of the acid chloride. So we used that for all the amino acids and all the carbohydrates, and then we could present that in 2001 in San Diego at the peptide meeting, Wave of the Future. So what is it? Click chemistry is this sack of electrons. It's the high electron density here in the center of the triple bond. It looks for a positive charge, and for some reason, copper 1 is extremely good at binding to the alkynes. So it binds here. This acidifies because we are taking the electrons away here. So this becomes acidic, and it's very easy to take off and replace with a copper. So now we have another molecule, but it's just it looks exactly the same as before, except now we have a copper there. So that's not very reactive, but if we take another copper atom, it still finds the center there, and then it peels off the electron density of the alkyne to leave us with very high electrophilicity at this center. So that's what that looks like, and this is the LUMO of that compound. That means the empty sacks. No electrons in there, and then we come with full sacks over here. These are the orbitals on the A side, and you can see how beautifully they fit in phase and geometry to make a 6-membered ring. So here and here we have overlap, and we form 2 new bonds like that, and then it loses 1 copper atom, then a second copper atom, and we have the triazole. So now Christian has connected a phenyl group and a metal group, and you can ask yourself if that is exciting. I would say it's probably not so exciting, but the fact is that, we have just one problem in this reaction, and that is that copper 1 tends to go to copper 2, which is completely inactive. There's a very low barrier for this conversion, so any oxygen present will actually do this reaction, and that wouldn't matter because we could just reduce it back. But this is a problem, because when we do this oxidation here, we get all the radicals, and you can see what happens if you take a macromolecule of biological origin. Here you get plus 16, plus 32, 48, and so that's these radicals that react with the protein. So ROS is destroying our molecules, and we have to remove the oxygen completely and not rely on this reduction. In order to get it to work properly, we need to have a high enthalpy to make it quantitative, and also orthogonal click functionalities like the A site and the alkyne that doesn't react with anything else in the biomolecules, but it does react with each other in the presence of copper. Easy access and easy to install, selective, biocompatible, and clean reactions, stereo and radio-selective, and most importantly, it's a catalytic triggered reaction, so we can add the copper at any time during this to get the reaction to go. It's insensitive to carbon dioxide and water, but not to oxygen, and that's the only critical point of this reaction. It's coordinative in a transition state. That means that it, at very low concentrations, can assemble the reaction partners on a copper cluster and make the reactions go at physiological concentrations. So this is how we do it. We have a click precursor molecule, and we inject it into the click mixture here. So that is in particular important if we want to cyclize molecules so they don't get to long linear compound, but actually cyclize. Then we do it very slowly, add it here into the mixture. But click reactions, because they have to fulfill all of these, are really difficult to find. You only have the TCO reaction, the QAC reaction, the spark reaction, and a few other ones. Maybe the SNAR reaction from pentalute can also be called a click reaction. So here we have three components. We have a killer, we have an entry component, and we have a locating component. And these are supplied with click residues, either through synthesis or through amber stop codon expression, if it's proteins. And then we can mix these three components in a container. They steer around overnight, and we isolate a single molecule that can then be used, for example, in recognition here of a cancer cell. Boom, and then it can enter via this ninja-like moiety. Boom, goes in, and that actually kills the cell through interaction with the XEAP, an inhibitor of apoptosis. So click chemistry can be used for these kind of robot molecules. But there's so many other things that can be used for, like ligation, peptide stabilin, surface immobilization, polymer synthesis, and so on. So that's why it became so broadly utilized. I will give you some examples of our use of click chemistry. And this here are some guys that are 400 million years old as a species, and they have a pharmaceutical factory in their blood. They produce toxins like this one, and macromolecules that can encapsulate foreign organisms. So we actually harvest the blood of these individuals for monitoring bacteria in human blood. But we were more interested in the toxins. This is targetplacin 1. And here we have targetplacin 1 with 2 disulfide bonds. We replace those with triazoles, and this is work by Kai Holland-Nell. And we discovered in the process that they form dimers in solution, so that's why they are not toxic generally. We have these benign dimers looking like that, with arginine sticking out, floating around. And then when they hit a negatively charged cell membrane, they combine there and form a pore. And this pore is allowing all the interior of the cell to slowly escape to the pore. And the cell, of course, cannot survive that, so the cell will eventually die. We tried out our molecules on 4 different species, 4 different cells: coli, Staphylococcus, Salmonella, and Bacillus subtilis. And we ask ourselves, is our compound as active as a natural targetplacin? And yes, they were. This is targetplacin. This is one of our compounds, and this is another one of our compounds. These sort of beta hairpin-like molecules with triazole bridges. And you can see one thing here. They are evenly active, but you can see here there's a completely inactive target placing activity in Bacillus subtilis, and our compounds are fully active. So probably the Bacillus has a way to reduce the disulfide bonds and make this an inactive molecule. This is from the Nobel ceremony. And this is the first day in the museum. And here you deliver one thing that has been very important for your research, and is now in the Nobel Museum. And this is a small block with 20 columns where we did our first combinatorial chemistry in 1988. So these were multi-column systems that could be used for a lot of different things with solid phase synthesis. And they are now in the Nobel Museum. And then you have to sign this chair in the bar. And I'm very proud because this is right next to Emmanuel Carpentier and Benjamin List and other people are on this chair. So molecular recognition, as I said, is a very important thing. There's a molecular recognition event in front of any biochemical reaction that happens in you all the time. So two molecules come together and recognize each other specifically, and we thought antibodies are the primary recognition molecule of nature. We could make that much simpler in chemistry by making these small beta hairpins inspired from the previous project. Here we have a beta hairpin where the structure is maintained by beta branched amino acids on the top, the turn here, and the bridge, and we have variable residues sitting here on the lower side. So we can actually take these into the computer and completely design a recognition molecule to any specific site on a protein. We have done that here. We designed two molecules, this one and that one down here, and this here is interleukin 6. And you can see they fit very well in shape and charge complementarity on the surface, and therefore we get high affinity. So we have one of them attached to the beads and another one in solution with a fluorophore on it. So when we mix the two, nothing happens. But then we add 20 nanomolar interleukin 6, and you can see immediately we get highly fluorescent beads out of it, meaning that this one recruits interleukin 6 and that one binds to the opposite side. So we have designed two recognition molecules in one go without any problems. That's really promising, and we thought we could take that into a more useful aspect. And here, Greener at Allied have isolated fibrils from a plaque. This is a plaque in an Alzheimer brain. And these fibrils were isolated, and by electron microscopy, you got the structure. So we took that structure, and we cut it in two here, and got the two growth phases for the fibril formation. And we designed molecules that bind either to the top side or to the bottom side. And this looks like that, two molecules. And we have here a fluorophore that inserts into this when the fibril forms and fluoresce, and that's the fibrillation curve here for a beta 1 to 42, boom. And when we add our beta bodies, we have a little bit of fibrillation here in the beginning, and then it dies off. We're not completely happy with that. But what we also observed is that even if we let the fibrils form first and then add the peptide, we reduce the amount of fibrils very significantly. So that here is an opportunity for dissolving preformed fibrils. So we were not happy, as I said. So Yan Zhang was a PhD in my group. He designed these molecules from glucosaminic acid, which have the same structure as threonine in this part of the molecule, as D-threonine. So he introduced a protecting group, first an F-MOC, and then TES, so triethyl silanes on the oxygens, and then he removed the one on the carboxylic acid to get this compound as a building block for peptide synthesis. So very important, it has the same configuration here. So we can use that on the structural side of these beta bodies that we call beta bodies. And here you see one of those with the glucosaminic acid sitting there, or here in 3 positions. We took these peptides, 1, 2, 3, 4, 5, and then we tested them against the fibrillation. So here you have 1 to 42, here you have 1 to 40 Alzheimer peptides. And when we mix the different peptides, you can see different levels of fibrillation. When we mix 3 and 5, so these two here, you see no fibrillation at all, neither here nor here. So that was extremely encouraging. The thing that happens is that we add a water shell around the glucosaminic acids that helps the recognition, presses in against the recognition partner. And it looks like that. So that's actually our molecule that we have designed. And this is what it looks like when it takes out small fragments of fibrils. And our hope is that you can wash out these small fragments during your deep sleep at night, together with all the other debris from the brain. We don't know yet. During this program, we also looked at the possibility to cleave Alzheimer peptides in the nucleation site, so they wouldn't be able to form fibrils. And we made a small substrate of this fluorescence quench substrate of the nucleation site, and then we made carbines in peptides that could bind to a transition metal, so this could catalyze the nicking of the Alzheimer peptide. We also bound them to bipyridyls, and then we combined that with all sorts of metals, and it worked. It worked fantastically. And it worked in all cases, both this one and that one, and all the metals worked fantastically. So we were a bit suspicious here, and Martin Wolfram, who did this work, he took out first the metal, still worked. He took out the catalyst, still worked. So what is going on? We have activity even without these organozymes present. So what is going on? And that's where you have this moment of observation where you think, well, this is unusual, so maybe we should investigate it further. And what we saw was this is an autocatalytic activity. We synthesized all of these fragments of the peptides, and we investigated everything that happens here. And this is, for example, a peptide where we start just making a solution of the peptide. After 11 days, we see something else formed, then something else, something else, and something else. And we can isolate all of these and show by MS what it is. So these are the fragmentations that we are observing, very specific fragmentations. And why is that? Why does this happen? And why does it happen only after 11 days? So we also took some of this and added to a longer beta peptide. And here you see, after a few hours here, it suddenly starts to cleave. So what is happening? Well, we also took the full length 1 to 40 here, which is in solution, and we seeded it with that one, and then, boom, after 12 days, you get this. If we don't seed it, nothing seems to happen. We thought, okay, let's try it with 1 to 42 and just leave it and see what happens. So my student made a solution last May, and then she came back in June and measured, and nothing happened. July, nothing happened. August, nothing happened. Then in the middle of August, she did another assay, and suddenly it looked like that. So again, a very delayed autocatalytic hydrolysis of itself. That is a pretty strange thing. Could be a precursor to a lot of enzyme activity in biology evolution. Another thing that we observed was that these forms initially these loose donuts of single-stranded molecules, and they then collapse into small strands. You can see the collapse several places to small sticks, and then these sticks combine into long fibrils, and the fibrils mature to get you mature fibrils like this one. So we have a mechanism for the formation of fibrils. We have an intervention in their formation, and we also know that there's a catalytic process going on. What is very important is that in the presence of Alzheimer peptides in the brain, in the vicinity of plaques, we have neuronal cell death. And this is caused by cleavage of the NCAM2 molecule, which is the molecule that adhere the neurons together so that they can actually communicate and form the correct contacts to communicate. So if we remove the NCAM, the neurons, they cannot communicate and they actually die. So here you see a normal brain, and we see very little here of the soluble NCAM, and we see a lot of the full length. In the Alzheimer, we see a lot of the soluble NCAM, and we see also some full length, but not a lot. And it spreads out like that. So this is the normal, and this is the Alzheimer brain. And also, this seems to correlate with a cleavage in here, because we can see that from the isolated fragments. And what happens is that actually co-localize, this soluble fragment co-localize with the Alzheimer peptides. So that's very interesting. Also, the sequence of the Alzheimer peptide that cleaves is here. And here you see the cleavage site for the other protein, very likely. We also like that there is a very long initial time before this aggregate forms, because that means that we can live a number of years with Alzheimer without any problems, and suddenly it happens. So this is what happens. Our aggregate of small peptides that have catalytic activity comes in, adheres here, and cleaves off the NCAM, and of course this is not good for the neurons. So we have a mechanism, we have the formation of the donuts, and we have a catalytic entity. We are back in Stockholm, and this is the day of the presentation. You're pretty nervous, you're talking to 10 million people. But it went well, and here we are, the three of us, all very happy about the result. So I would also like to present our work on import into cells. So cell penetration is a very important thing for pharmaceutical industry in particular, because most of our targets are actually intracellular or across the blood-brain barrier. So this here is our key molecule from nature, the HIV TAT peptide, which has some import. And based on that, there's a lot of arginine residues and lysines here. We synthesized and designed this small molecule. This is 1 out of 16, as you will see, with positive charges and a hydrophobic part on a proline turn. So that actually works quite well. We have to synthesize first the molecule, so we have a protection of hydroxyproline. Then we adhere, or we alkylate this position with a long fatty chain or a aromatic chain. And then we reduce off the protection here, and we have then the building block after FMOC protection. We can also do it by double inversion to form the aminoproline, and then we go all the way to FMOC, BOC protected, and then with an R group that is hydrophobic. So we have these 4 building blocks, actually 5 building blocks, that we are using in construction of peptides. These are the 16 cyclic peptides that we made out of those, 12 cyclic and some linear. And one of them, this one here, was extremely active, as you can see here. Maybe you can't see it, but anyway, there's really nothing in all of these 16 except in this lane here, where you can see very clear import. If we look at that in more detail, it looks like this. Here the import, and if we just compare this with the same concentration of TAT peptide, you can see there's much less than up here. So we have a very good peptide for import. This is sort of the quantification of these slides, and I think this is actually much lower in reality if you look at these images. We can synthesize these molecules very cleanly on our synthesizers, and this is a TAT peptide. We also synthesize that, and you can see here the masses. And we use this like standard equipment that we build ourselves. Equipment is expensive, so we build our own machines. I've just made 7 of them for the students, so they can all make these kind of peptides. We can investigate the process. Here's the temperature dependence, showing that it's an energy dependent process. We can also see that 12 hours is the optimal time for intake. And we can see that we can actually observe fluorescence in the cell already at 100 nanomolar concentration of this peptide. We can also see what is the mechanism by using different types of inhibitors. Here we have the inhibitors, and dinosaur is the one that really reduce the intake, and that means that we have a dynamin-dependent endocytotic pathway, both here in MCF7 and in HeLa cells. We can also use this peptide in all sorts of cell lines. Another huge difference, all of them take it in. Here's shown the results from this assay. So we synthesize the peptide with a linker and an alkyne on it. So first have one click reaction to make the cyclic peptide. Then we have a second one out here to attach it. And we attach it now to green fluorescent protein, right? So green fluorescent protein here. And then when we take this green fluorescent protein, nothing goes in. If we mix that in solution, these two, nothing goes in. But if we have them connected, you can see very clearly that it goes in to a large extent. So we can actually import large proteins using this cell-penetrating peptide. And the way it works is that these guys, they find the membrane, it sticks into the membrane, and then it forms a vacuole that clips off and goes into the cell. We have also made alkylations of the arginines. So these are the arginines, and this is the way they are organized in connection with the phosphates in the cell membrane, the arginines of the molecule, and here you see the molecule sticking into the membrane. So we have also alkylated these arginines with either methyl groups or butyl groups, 16 of them, and then that changed completely the picture. Now we get not these endosomes, but rather complete distribution of the fluorophore in the cells. Here we are back in Stockholm. This is the day of the award. So here I say hi to the king and get the medal, and I'm on my way to the seat with the medal, and then somebody comes in and takes the medal away from me. So I thought maybe I said something wrong to the king. But I didn't. It's simply because many of us are much older than when we made our discoveries. And some of us has actually been attacked by Alzheimer in the meantime. So a lot of the Nobel laureates forgot their medals in the ceremonial hall. So now we get it on the last day, and then it's your family that makes sure they get it home. This here is an enzyme, and enzymes are notoriously, in particular proteases, are notoriously difficult to express because they are, of course, proteolytic, so they will proteolytically cleave the proteins of the host cell. So you can see how sad the coli is here. So we thought, what can we do? Well, we can cut it in two and use the amber stop codon technology to introduce 2 click residues and then express them separately, the 2 parts. And now we have these 2 parts that come together. We asked the coli, are they happy? And they were very happy because we got hundreds of milligrams of these 2 molecules. So here shown in more scientific detail what we did. This is a shorter bridge and this is a longer bridge that we incorporated. And we tried both of those. And you can see here the fragments. This is one of these fragments. Looks like that. And this is now the product after the click reaction. Here's a gel. This is the clicked one. This is the unclicked material. And this is a shorter bridge. With a longer bridge, we transform the one-to-one mixture here completely into product. So this shows the potential of this click reaction. We made these two. We coupled them together using copper 1. Here it comes. Boom. Get a triazole. And then we ask ourselves, is this enzyme actually as good as the natural one? So we cleave this substrate at that position, follow the fluorescence from this thread, and you can see the upper one is actually our compound, which means that our compound is a little bit more active than the natural one. So we were very happy with that, and this could be a very good entry into expression of proteases. We're back in Stockholm. And this is a day where you have the ceremonial dinner. And on the first day in the museum, my wife has spotted this dress on a mannequin. There were 6 dresses for the Nobel Prizes, and it looked beautiful. So she called the arts department in the science and arts of Stockholm University, and they said, yes, we can make this dress for you. And they did in 24 hours, and here she is, very, very beautiful, in this cover dress. But when you dress up your wife this beautifully, there's a real danger. And in this case, the danger is called Prince Philip of Sweden. And here he is. He took my wife, and I was sort of guided in this direction. And she was seated in the center of the hall between Prince Daniel and Prince Philip, and I was down in the draft. So, but it didn't really matter, because the next day we were invited as an intimate dinner at the Royal Castle, just 130 people, and I got the hot spot. So the last thing I would like to talk about is a different kind of click reaction. Here we have a reaction that was again discovered as a serendipity. We had tried to make N-terminal aldehydes or peptides, and it really didn't work for us because the aldehyde was doing something strange. What happened here is that we have here an aldehyde, and when we release that aldehyde, by removing the Boc group, this cleaves off, make an aldehyde, it actually condenses here. So we get the aldehyde condensing to hydroxylactam. This hydroxylactam loses a water molecule, make a very reactive N-acylaminium ion that then can react with nucleophiles in the peptide. This is a very cool reaction because now we can take a peptide and make small molecule out of it. So we tried that. First, we built all the building blocks that you can think of here by a double malonic ester synthesis, introducing first the aldehyde, and then here the benzyl group, for example, or other groups here in the side chain. And we also made acetic ester synthesis like that. So we had a variety of different compounds available as building blocks. We can also use alcohols to oxidize first as a ketone and then as an acid. We could also oxidize double bond, a very useful reaction, and then we can convert amino acids into aldehydes at the C-terminal, and then we can make the coupling via a urea at the N-terminal. So here's shown what happens with these nucleophiles and this arrangement, and you can see all of these reactions performing extremely well according to these HPLC. So this is really an intermolecular click reaction, completely quantitative, nothing else forms, only one stereoisomer. We can also see the crude NMR spectra. This is a dream. Usually it doesn't look like that when you go with your crude product. And We tried that with, so this was all very reactive C nucleophiles. We tried it with a variety of different C nucleophiles. It even works when we have 2 electron withdrawing substituents on the phenyl group here. Very good reaction, a single product. And you can see all of these compounds works very well. We can also use other types of nucleophiles here, oxygen, sulfur, and nitrogen. And that forms a different type of heterocycle here. You can see that is from homoserine and so on. So we have a lot of different N and C nucleophile. And if we don't have a nucleophile in the side chain, it just finds the next amide down the chain to form these kind of molecules. Amazing reaction. We can even do it inside a peptide synthesis. So we can start out here with this allyl glycine type of reagent. We make this condensation. So here we are synthesizing a peptide, this peptide here, also shown here, which is a mu receptor agonist. And the way it works is that we introduce this one here, and then we can continue the peptide synthesis to the end, and then form the cyclization by aldehyde, reaction here, and reaction with this side chain to form that one, 81% yield. Or we can go the other way and cyclize first and then continue the peptide synthesis to form the same in the same yield. So this reaction is completely orthogonal to the peptide chemistry. And we can also turn it around, make the aldehyde out of the amino acid, and then protect the amino group as a carbamate and introduce that to the amino acid there, and then we can get this kind of heterocycle. This is shown the result of that cyclization in a crude product. And we can also see that these products are very special because they can oxidize here in the center, and then we get very, very fluorescent compounds. So that is a way to make fluorescence in situ in a peptide. So we have really a combinatorial dream landscape here because we can use all sorts of nucleophiles. We get 100% stereoselectivity. We can use 5, 6, or 7 membered rings here and 5, 6 membered rings here. We can have different types of interaction with the backbone as well. So there's a lot of creativity possible here, and these are some of the compounds that we have made. And you can see this is a goodie box for pharmaceutical industry. A lot of these compounds look like pharmaceuticals, and they are out of the flat landscape, or flatland. So the world is an increasingly dangerous place. We see wars all over the place now, close to here as well. And I think we need to prepare for a future because of the strain that is put on human beings with the global warming and other problems. So we are lining up behind strong persons, and these persons have a problem in that they are autocratic. So we get a world that is ruled by narcissists, very important, because they engage in senseless wars, and they don't care about empathy or about global warming or about the community as such, only care about themselves. So we will have ice melts, plastics in the seawater, pandemics, flooding, fires, desert diversity loss, and so on. These are really serious issues, much more serious than the price of gasoline. So we have to prepare for it, and the only way we can prepare for that is via education. Understanding what is going on chemistry-wise is very important. So I think that we should actually train the kids. Here is a suggestion that we make a repository of videos, VRs, and other materials that is visual, doesn't require a lot of formula and math, just videos that can tell us about this three-dimensional world of chemistry. Now we are a teacher and we can call into this AI, and then you tell them where you are in your education, and you get the material necessary. This is very easy to globalize, so we can make it in Danish, take the sound off and make it in Turkish or Chinese or whatever. So we have the possibility to make a globalization unit here. I think this could be very important for the environment, for climate, global politics, for conflicts, including religious conflicts, for resource distribution, for equality in the world, and for wealth and peace. This here is what I suggest we do with the kids. I have sent in a patent formulation of a molecule that behaves like a real molecule when you build it. So a building sand with small magnets for hydrogen bonds and so on that really behaves with the resistance to rotation across the sp3 hybridization and so on. I think that would be really good for the kids to understand how molecules actually behave. And these, they have no language. They don't understand what is going on. So they, on the other hand, have this incredible image processing. So teaching them how to dive into a glass of water and see all the hydrogen bonds, see what happens when the water evaporates and when it condenses again or get frozen into ice as a starter. So we're creating a navigation mirror of the objective world. So we want most of our disruptive discoveries to really make a difference. In order to do that, we have to allow serendipity. And you can just look at the Nobel Prizes. 80% of them are completely serendipitous discoveries. And we need, of course, to make our research funding also increase productivity, quality, creativity, responsibility, and translation into industrial products. That comes all by itself. So I don't think we should do like we do today, put all the money for research into one big pile and then divide it into applied and so on from one big pool. We should actually define what is necessary for the research for tomorrow, what is necessary for the research that transform already known knowledge into useful things. These are two entirely different processes, and they should have their separate financing environment. When you do that, and when you focus on education, you can see here Finland, South Korea, Poland, and Vietnam really had a step forward when they introduced education. Education is the best return of investment you can make for society. So the conclusion is that education lead to broad long-term economic benefits. That is why it's so difficult to get it done, because in politics and in industry, we need short-term. We need short-term successes. So if we do that, maybe we can get some of our young students like you to be concerned with the global warming and with sustainable chemistries like transformation of water into hydrogen and oxygen, like new materials. This is a biopolymer that we made out of barley. It is just as good as plastics encaging your computers. And here is a catalyst that we made. This can be used for proteolytic activity, and it's completely synthetic with L&D amino acids. You can also think of foods, and in particular the oceans is underexplored for food. We should not be indifferent to the plastic pollution of the oceans, because this is a huge potential for food in the future. Textiles is a real problem in the world. Find solutions to the textiles, find solutions to transport, not this one, but better solutions. We should have a very promising future, but we should not forget fundamental research in molecular recognition, processing, and signaling, and also materials. So this is best done with excellence. Excellence is not a single person who's excellent. It's actually an excellence center. And I hear that here at METU, you have these excellence centers. I congratulate you with that transition, because I think it's really, really important that you have the support of excellent persons in their own environment. So you have a playful team of well-equipped people. They have freedom for all, not only for the PIs, but also for the postdocs and the PhDs in the group to actually go and do what they think is most important. So you then get research that is driven by curiosity, not by anxiety. I think that is very important. And then you get a true engagement. So you're not thinking, what is happening with me? You're thinking, what can I do tomorrow to solve this problem? And then you're allowed to do so. So you get the independent co-workers in the university that really establish this environment, this future of brilliance, which is going to go into the society, into the industry, and so on. And with that, I would like to thank METU for this opportunity, thank the Danish National Research Foundation and Carlsberg Laboratory and University of Copenhagen, Claus Bock, my former supervisor, Michael Bolz, the director of the Chemistry Institute at Copenhagen University, and Carlsberg Workshop, because they have really supported me with a lot of instrumentation. I would like to thank my collaborators outside and also my co-workers at the group of Carlsberg. I would also like to thank the people at the Copenhagen University. These are all postdocs and PhDs at the university. And this here is my family, Phaedria, Gianni, Anna, and Sandra, that has been very supportive, not only allowing me to, but actually contributing with scientific discussions over dinners and in the laboratory. So thank you very much.
Thank you very much, Professor Meldal, for this truly inspiring and insightful lecture. We will now move on to the Q&A session. It is my pleasure to introduce Professor Özdemir Doğan from the Department of Chemistry, who will moderate the session. And for the Q&A session, if you would like to ask a question, please feel free to raise your hand. Our student assistants with microphones will be circulating around the hall, and we kindly ask you to direct your questions through them. And now, hand over to the Professor Özdemir Doğan.
Do you think you can find some water? Tell her I got water. It's okay. Sit down.
Professor Meldal,
Yes.
first of all, thank you. It was a great lecture.
Thank you very much.
You told us and showed us how great is the chemistry in
Yes.
terms of recognizing molecules, in terms of recognizing cancer cells and all kinds of biological activities. And also your last message about the leaders and the world,
Yeah.
and also how we take care of our kids and how we educate them.
I find that we as scientists are really responsible in that regard.
Yes. Thanks a lot. Thank you very much. Yeah, please sit. Maybe I take the advantage of being a moderator of this session to ask first question. We have a saying that there is always a woman behind a successful man.
Yes. We have that saying
Do you
too.
agree?
And in this case, it's actually true.
Yeah. Thank you, Maria. Maybe. Well, I also thank Dr. Erhan Atıcı for organizing and bringing Dr. Meldal and his wife to Turkey and to our university. Thank you very much, Erhan. And I also thank Eren Hoca. He organized everything, Eren Hoca and his team. Thank you very much for your efforts and all your work. Thank you very much. And now I think we are ready to take, well, I also thank, of course, all of you for participating at this wonderful event. So thanks a lot. And I think we are now ready to take questions for Dr. Meldal. Who would like to ask the first question?
There's no stupid question, so here we go. There's one over there.
Yes.
Please take that one.
I got the microphone, by the way.
Oh. Yeah,
Microphone is
okay.
there. Professor Meldal, firstly, thanks for your presentation and sharing your experience to your colleagues and to the students of the university. As a material science and nanotechnology student who has also studied organic chemistry, I am fascinated by the elegance of your discovery. Today, as you know, we can see AI-driven retrosynthesis and high-throughput screening tools attempting to map the entire chemical space. In your opinion, do you believe AI can eventually predict or invent new click-like reaction archetypes that go beyond human chemical intuition? Or is this the discovery of such a fundamental chemical beauty like the triazole formation, something that requires a uniquely human form of molecular creativity that algorithms that cannot replicate?
Yeah, so I think there's a difference to intelligence and AI. And the difference is actually in the intuition, in the ability to create things that are completely non-existing in what is already there of the human brain. I think that AI will improve on that with the development of chips that are simulating the structure of the human brain, because that is what is required. Behind that, there is also necessity to develop a consciousness that is behind the decision, the bridging of associations and so on in this artificial brain. So yes, I think that artificial intelligence will eventually be competitive. It is not at the moment. Actually, there's a huge problem with artificial intelligence in that for chemistry, the publishing houses are sitting on a lot of the data that the AI needs, and you cannot rely on the AI to have access to all this information today, because, of course, the publishing houses need to protect their property, and they are doing that very firmly. So you need some sort of agreement between the publishing houses, the governments, and the academic institutions where AI can get access to all those data. Because today, if you try to get a kind of reaction, I tried that many times, you get some suggestions, but they are, if you are expert in the field, you can see they are not always the best. They don't include all the knowledge of the literature. But there's other areas where AI is perfect, like in the crystal databases used for AlphaFold. AlphaFold is amazing. I think that we have many times, again, produced results that are very reliable using AlphaFold. The good thing about AlphaFold and the way it's constructed is that if you don't know, it don't tell you. So that is why you are very sure about the structured data you get, and it's much better than a human being in filtering the results. Any more?
But thank you. Next question.
Did you hear me?
Yes.
Okay. I'm wondering, what was your dream to have a Nobel Prize when you were young?
Not at all. Actually, no, but truly, I've always been much more, as I said in the video before, I love to be in the lab. And this is why I became a chemist, is because I love to be in the lab. It's like a sandbox of tools and things and shapes you can make. And it's wonderful to be in the lab. I love to do a chromatographic column and get a pure compound out and go to the NMR machine, get this wonderful spectrum of the compound that I made, you know. That's really, really nice. And I think that's what is lacking in many ambitious scientists, this feel for the actual chemistry, not for the ambition of being a Nobel laureate in the end. So it came to me as a surprise.
Actually, it was my dream for 40 years in Turkey. I want to be first Nobel Prize in chemistry. I am organic chemist and medicinal chemist, but not anymore. For 40 years, my dad's final decision is not possible in Turkey.
No,
so sorry.
I mean, it is possible, but it's not coming to you because you want it. It's coming to you because of what you did. And Denmark is a very small country. It's not every day we get a Nobel Prize in Denmark.
I know. Your country is
6 million
people.
6 million people.
And you have 14 Nobel Prize.
Yeah.
We are 85 million.
Yeah, so we were lucky to have
one.
Niels Bohr and his guys in Denmark, yes.
Big science needs more money. I'm wondering how much big money you can get for your research. What is the research found in your country?
Yeah, so we have a very special situation in Denmark because of the high taxation, which we love. We think it's very good to have a high taxation. And actually, it's a conundrum for the Americans that Denmark is one of the best economies in Europe with the highest tax rate, you know. That's not possible. That should be a no-go for a US economy. But it is actually one of the best economies in Europe. And the reason is that the taxation allows the companies to invest their money there. Instead of paying taxes, they can put the money into a foundation. And this foundation just have to support societal issues, including research. So if you are a company in Denmark, you're doing very well, you will form a foundation in order to avoid taxation, and then you will have to pay back to the society. And that's a really, really good model. So Novo Nordisk Foundation is supporting with more than the Danish government in Danish research.
Okay, can you give us the money, what you have? What is it? How many million euros?
So in euros, I would say probably over my entire career, about 10 million euros. Not
good.
more than that.
More money, more science, more Nobels.
Yeah, but these are, I mean, so this was foundation funding because we decided, for example, to go into glycobiology, glycosyl amino acids, glycopeptides, glycoproteins. So we got from EU, because that was a new area, so we got from EU some funding for that. Then it was the solid phase organic chemistry that you saw here today. That also got some money from the Danish National Research Foundation, about 4 million euro total over the years, so that was over a 10-year period. But we haven't had that, you know, huge funding that you get in the US.
Okay,
Thank you.
Professor Vildan
Well, we have
Hacettepe
other
University. Thank you very much.
questions, please. Thank you.
Thank you for being here. I was curious about the water layer that you have shown as the inhibitor and the curiosity about the water. Can you comment on that a little bit?
Yes, so every molecular interaction is a very dynamic process. A receptor and a ligand that has to find that receptor can only find it in one way, and that is by an energy-driven transport towards the receptor binding. That happens through the network of hydrogen bonds in the water. So you would eventually get to the minimum energy situation, which is the bound ligand and the water surrounding it. So water is a very important feature. It's actually the most important feature in biology, is water, and all the connectivities that water does. So water is actually acting as a hydrogen-bonded network on the top of the molecule because we have these polyhydroxyl groups sticking out. That's the same that happens in a glycoprotein. That is what makes some glycoproteins soluble, for example, or that they fold in a particular manner. Because the water interact in the local environment of the protein, the water interact to solubilize it, to fold it in a particular way. And we have the same situation.
Yes, please.
Thank you very much for the inspiring talk. This is Gürkan Yeşilöz from National Nanotechnology Research Center, UNAM, Bilkent University. So we are located in this Ankara city as well. So I have one technical and one general question. The technical one is, do you see further advantages initiating these chemistry reactions in small scales? For example, utilizing microfluidics, which may enable them to control better the flow conditions, mixing or diffusive transport phenomena, which I'm working in that field. Can we increase the efficiency or maybe, create better reactions? This is the first technical question. For the general one, during your talk, you mentioned soft skills, some of them, but I would be more interested to hear further soft skills that you think, these are important for a scientific person.
What was that?
Soft skills.
Swap skills.
Soft skills, yeah.
Soft skills, yeah.
Yes.
Yeah.
Maybe, with the students here and the colleagues, if you can share further soft skills that you think these are important, that would be also great to hear again from you.
Yes, just
Okay,
so I'll come back to that. So with respect to flow technology, I mean, we are using flow technology in solid phase synthesis. We are using continuous flow synthesis. But in the microflow environment, I think that is a very important one, because by flow control, you can control kinetics extremely well. You can have very dilute things coming together and react immediately, so you don't have, site reactions and so on. You can also produce quite large amounts in small flow systems, because you're continuously producing your product at the other end. So I think there's a lot of future for flow technology in synthesis. I'm not sure, though, that click chemistry is the one, because click chemistry is actually not fast. People think, oh yeah, click, but that's not the case, actually. Many cases, we have to wait 2 days before the click reaction is over. The trick with the click chemistry is that it is 100% selective, right? And it happens. So eventually it will go to completion if you are doing the right thing. This is the force of the click chemistry, not the speed of reaction. Sometimes it's over in seconds, sometimes it takes days, like any other reaction, actually. If you look at glycosylation reactions, you can have very reactive glycosyl donors and acceptors. They are over in seconds. In other cases, you have to keep the carbonium ion character for a whole week before you have the product. So this is of course, huge differences in kinetics. And flow chemistry is best with fast kinetics. Also because you can break the reaction after like 10 microseconds of reaction, you can quench, or you can add another reagent and so on. So you can really control things in flow chemistry. So I think it's an important field, and I think that it can be utilized for many, many different things, but it's not so easy technically to do that for every single laboratory in the world. Oh, the second one, the soft skills, yes. So the soft skills that I think is very important is the ability to imagine stuff in your brain. So do you have an imaginary ability? So I actually, when I have to hire a new postdoc or PhD in the group, one of my questions in the interview is, imagine a cow in a field under an apple tree, and then describe the rest. So they have to start with this picture that I give them, and they have to paint the hooves with lacquer. They have to make golden horns and flies around, and apples that are red and shiny on the tree, so on. So they have to make this image, the small stream that flows by, with the wetland vegetation and so on. They have to do that and make a good picture for me so that I understand they have the ability to imagine stuff. Creativity. So what kind of creative figures comes out of that? Are they able to actually create their own picture or their own way of seeing things? Courage. What have they done before? Have they dared to do things that their supervisor was not expert in? And so on. So you can actually test for all of these things. Have they written a proper thesis? Do they have this perseverance to work to the end so they get a good result? Are they good at searching? Are they good at associating their knowledge and do searches into the literature? AI, can they use AI? And so on. All of these are soft skills. It's not the actual knowledge, but it's building the imaginative universe of organic chemistry that's very important to me.
Yes, please.
I would like to ask a bit philosophical question. You said that chemistry is everything, and as we see in your presentation, it's mostly related with biochemistry, and we see that how chemistry makes biology tick or click. But physics people also say that physics is everything. And what do you think about this opinion? I just like to...
I'll give you an answer.
I just like to understand your opinion. How do you draw the border between physics or chemistry or biology or other domains of science? Or is there any border? Should we draw any border?
No, we shouldn't. Okay. And so I really think that we should try to look at chemistry and physics as the two descriptors of the whole. So when I say chemistry is everything, I don't think that chemistry is all. I just think that it's everything, right? And physics is all the energy flow, all the photons, all the things that actually happen are caused by energy changes, which is given by Gibbs free energy equation. A beautiful equation, the most beautiful equation in physics, extremely simple. And so understanding what entropy is and what enthalpy is extremely important in chemistry. Understanding quantum aspects of chemistry is extremely important. That's also considered physics, but it's actually chemistry, because it's how the electrons are moving around in the molecules and why they're doing that. So that's chemistry, in my opinion, but understood through formulas and so on in quantum chemistry. Science is a description of everything. Chemistry is everything.
I was going to ask about your research. You were talking about how funding, like fundamentally, has to change.
Where are you?
I'm over.
He's on the right.
Okay.
You said how the system of funding and the project of funding should be changed into two sections for applied and fundamental. I want to know and understand more about your experience with the original or like the current state of funding and how it affected your research, or maybe how you felt pressured maybe to provide tangible results, and how it affected your ability to reach the discovery of click reactions.
So exactly with the click chemistry, we got the funding from the Danish National Research Foundation, which is the first of its kind to actually do an annual update of the goals of the research program. So every year they would come and visit our research center, and we would have to present, and if we failed to do that, we wouldn't get the funding for the next year. We would have to present 30% new ideas, never heard of before, which I think was excellent, because that allowed us to actually step out of the track and go towards these new discoveries that we saw. And that is what I think that then was adopted by the ERC later on. My former boss, Klaus Bock, he became actually both leader of ERC and also of not leader actually. He became part of the managing board of the ERC, but he also became the head of the Danish National Research Foundation. So after we got this grant, he actually got into these situations, and he copied what was in the Danish National Research Foundation into the ERC. And I think that many foundations are actually now looking at this as, how do we actually make breakthrough research?
Yes. Peki, oraya alın.
Hi, I'm Hilaire. I am an assistant professor at Hacettepe University. Actually, last 12 years I was living in the UK. I have just moved to Turkey last year. So previously I was a postdoc at University of Cambridge. It was such fascinating talk. Thank you so much, Professor. But my question is a bit political. I think I asked these questions to other Nobel laureate people as well. So what do you think about the next Nobel Prize subject will be?
I don't think that should be a political question. Hopefully not.
You know about Donald Trump, that's true.
So this is, there are so many different areas that deserve this. I think that one of the most important things right now is to understand the energy. How do we actually, by chemical means, handle the energy issue, the energy situation? So that's not only from a practical point of view, but philosophically as well. And I think there are some groups around the world that are working on direct transformation of photons into hydrogen from water, which is a very likely subject for a Nobel Prize, in my opinion, depending on the success rate, of course.
Thanks so much. Thank you.
But that's just one. There's so many out there, and I cannot tell you what is going to be the one that is selected.
Yes, please.
Hello. Can you chemically target, I'm here. Can you chemically target the macromolecules into nucleus?
Not with the current technology that we have. It doesn't go into the nucleus. We would have to add a nuclear pore transporter to our cell-penetrating molecule in order to do that. So you have to have something that allows it to interact. Actually, we deliberately didn't want it to go into the core because we have previously made cell-penetrating peptides with lots of positive charges, and those actually go into the nucleus in order to interact with the phosphates of the DNA. So if you have too many positive charges, it becomes toxic and interacting with the nuclei. And we deliberately try to limit the positive charges as much as possible. Yeah? So we don't want to target the nucleus initially, but for a particular purpose, if we have selective recognition partners that can transfer into the nucleus, that's perfect.
Okay.
Yes, please.
I have also in fact, few questions. One is, before getting the Nobel Prize and after, how was the situation about the fundings that you were receiving? Did it change the behavior of the, foundations to you after you get the Nobel Prize? And did they maybe more willing to give you, fundings?
Yeah, so of course it has an influence. Yeah. So what happened is not that I suddenly got through with my applications. What happened was that they both recognized this honor for Denmark by taking out two of my own, so two foundations, Willem and the Novo Nordisk are the two big foundations in Denmark. Both of these took out an application that I already made and was rejected and looked at it and said, maybe it's not that bad anyway,
Okay,
so
quite positive.
So I got a grant from each of them, but it had to be a very small grant because if it's too large, it has to go through all the panels and they have to go through all of that. They wanted to just give me, so I got about €1 million totally just to continue research, and it's been fantastic because it was not associated with any program. So I can use the money as I like to play around in the lab.
Yeah, exactly. I believe that organic chemistry is not an expensive science because it's, mainly regular, instruments, we are using. What can you comment, to the society? Because many times we hear that, we don't have sufficient funding. Of course, we need money for that research too, but compared to other big molecules like biotechnology and such things, compared to them, what do you think about the cost of, studies in organic chemistry?
Of course, it helps to have a very large instrument that can give you accurate data and so on. But a lot of the things that we do can actually be done with much less. Yeah. And I don't think that it's not an excuse, in my opinion, that you don't have these large instruments to make good research. You can still make good research. Personally, I just built 7 machines for, 50,000 euros. 7 machines to build peptides in the lab for nothing. Yeah. I used the Raspberry Pi computer, touchscreen and stuff like that, and build a machine that each student has one, so they cannot complain. They just have to do the work now, and it's really working well. I have my HPLC, the PrEP HPLC. The controller there is 35 years old, still working fine. You can put together scientific instruments if you want to. You don't need to have the most expensive on the market.
Yeah, that's great to hear this, from a Nobel laureate, really.
Yes,
One
we
small
have
other
just
question, and I leave
not
it.
related with funding, please.
No, not related to money. But I would like to hear also, since you are working on peptides, there are now very good, effective GLP-1s. In fact, we discussed a little bit. Are you working on new GLP-1s together, maybe Novo or yourself in lab? Thank you.
No, so our work is mainly related to the Alzheimer, to oncology, and to viral infection, stuff like that. We don't have anything on GLPs. Interleukin-11 is another area that we work on because of its health, relationship to health, in particular, late stage in life. So these are the things that we are working on. Not so much weight loss, because I think a lot of companies is in that area, and they will do much better than I will.
Yes.
So first of all, I would like to apologize if my wording is a bit rambly and too long. So I generally am a person who wants to understand things intuitively as well. I tend to understand things with a level of, I think this makes sense with the current knowledge I have. But I have a problem going on with macromolecules, as they're so much more complex and there are so many more interrelating interactions between other parts of the same molecule. How come, or how is it possible that we can try to make some other molecule that targets something else specifically and maybe like predict, design a specific structure for the molecule that we want to create?
Yeah, so today we can do that, but only very poorly. So there's a problem. I think we do not completely understand molecular recognition. There's something that we are missing both in the force field and I think also in the quantum chemistry actually, that probably is associated with very weak interactions of electron donation into empty orbitals. But I don't know that. I cannot tell you what to do to find out whether that's correct. So this is just a personal philosophy of mine, that there is something we are lacking because we can calculate very accurately within a millimeter how to land on the moon, but we cannot calculate two molecules coming together. We can go a million wrong if we take an antibody and an antigen and try to calculate that. The affinity can be a million wrong in the computer compared to what it is in reality. So something is really, really bad in our calculations. Still, it works because the overall, we have the knowledge of electron densities coming together, a little bit of van der Waals interaction, which is also electron densities coming together, actually. And then you have the bond rotations and angle strains and all of that. Works well. The quantum mechanics cannot today, probably also not tomorrow, and probably for foreseeable future, calculate complex molecules in water. It's just not feasible.
Yeah, quick follow-up on that, because I do have the same philosophy when it comes to this. But if there was any way that I could better my understanding on this topic, what would you suggest? Since that's your work area, I wanted to know your opinion on this.
Yeah, so I think that we need to sit down with the theoreticians and find out how to incorporate every single interaction that is there, because of course it is possible to calculate this. It's even possible to do it in a force field format if we understand what it is. But there's something that we are lacking that we need to put into our equations in order to calculate it. We can still find something that looks perfect and binds in a certain area, binds with a certain affinity, and we can improve that by either medicinal or combinatorial chemistry. You can improve a little bit, and we can get nanomolar, even picomolar interactions eventually in a large research program. But it's not something you sit down in an afternoon and calculate on a computer. Something that comes, the high affinities comes from long series of optimizations. For drug candidates, it's not only about the recognition, it's also about the circulation time, it's about the stability, metabolic stability, and so on. So there's a lot of different things that goes into making new drugs that need you to make a multi-component approach.
Yes, please.
I'm chemical engineering undergraduate student and president of AICHE community at Bola Bandizat Basel University. So thank you so much. I want to thank you first for your great speech. And I have a couple of questions for you. The first is related to chemistry, has been compared with molecular Lego blocks together. So
molecular what?
Molecular Lego blocks together.
Lego blocks.
Okay.
Yeah.
Yes. When you first discovered it, did you anticipate it being used for target cancer and DNA, or you have been surprised by this?
No, so we developed the reaction because of the serendipitous observation. And we, of course, immediately saw that it's compatible with all the bio entities, so amino acids and sugars and so on. But we didn't target a click function at that stage. It was only later, like 2-3 years later, that we actually started to do that, making cyclic peptides, making surface modifications and stuff like that. So the click aspect grew slowly out of it. It was defined by sharpness at the same time that such reactions would really promote the Lego structure of chemistry, would really promote functional molecules. So that is why, so that's the great discovery of sharpness, is actually defining what click chemistry could do for chemistry.
Okay. And the second is, after you won the Nobel Prize, did it open more doors for you, or it add more pressure for the situation?
Yeah, when you get a Nobel Prize, you get a new job on top of your old job, right?
The pressure is going to be more high.
Yeah. So you have to disseminate your results. You have to keep the world going, you know, spinning. So you also have to do your research, and you have to do the teaching at the same time. So you're traveling around while you try to land running in Copenhagen and go to the teaching, go back to the airplane, go somewhere else, go back to the teaching, and so on. So it's a lot of work, actually, getting a Nobel Prize.
Okay, thank you so much.
Okay, yes, we have 2 questions. All right, okay, go first, you, yes.
Thank you. First of all, I am delighted to see you here, and I'd like to ask you this. As someone who wants to encourage young people, are you planning to organize another events with young people in Turkey? Because this really motivates us, and at least for me, I really inspired you from presentation and your speech. Thank you.
I have no immediate plans other than tomorrow. So tomorrow in Gebze we will give another presentation. I have not really decided yet whether it will be the same as this one or something different, but I will.
Thank you.
Okay.
As a chemical engineering who have an interest on sustainable catalysts and hydrogen system, what is the main industrial limitation of the click chemistry in terms of selectivity and production cost of these sectors? And how do you control the impact of the triazole linker on the physiochemical properties and of the final material, and its effects on material balance?
Yeah, so first of all, I think the scale-up problem really is associated with the danger of azides as an explosive material. If you have small molecules with azides, there's a real risk in large-scale production. If you have larger macromolecules with azides, it's not a problem at all. So there's a certain limit where you have a danger. So we had one postdoc in the lab who actually prepared hexaazidobenzene. You can imagine a benzene ring with 6 acido groups sitting around instead of the hydrogens. That was pretty explosive. But other than that, I think that this is not a problem to scale up. Azide is a very atom economy, positive entity. It's very small. Even if you release nitrogen and just use the amine, azides are perfect. In the click chemistry, you actually use all 3 nitrogens. You make a very benign molecule by reaction with an alkyne. All of the atoms in the starting material are in the product, so there's no waste, essentially. You use a catalytic amount, and we can go down to 0.1 mole% of copper 1 if we just maintain the copper 1 status all the time during the reaction. If we make sure we have no oxygen, for example, or have a good protective ligand around the copper. So it's possible to make these reactions really green, and you can carry them out in water. So scaling up in water is not a problem, right? Actually, it's a very nice thing to be able to do that. And if you have a quantitative reaction where all atoms are entering into the system with a catalysis of 0.1 mole%, you have a really good case in water. So I think that the scale-up for click chemistry is wonderful. It's also saving you in other ways, because if you're making a large molecule and clicking it together in the middle at some point is the key to the synthesis, you actually can do this as in a convergent way where you're making these 2 building blocks and putting them together, maybe even in water at the end. right? So this synthesis is more simple, this synthesis is more simple, and then you couple in water. Whereas if you have to make the whole thing through a large array of reactions, you have very little result and poor with a lot of waste. So click chemistry has a great future in that regard. And I think it's also very useful. For example, you were saying you're working in energy. I think that, for example, you can put energy reactive groups on surfaces using click chemistry, if you want to. We have done that, not with energy, but with other things. Put it on surfaces, on nanosticks, on indium and gallium nanosticks on surfaces, arsenides. We have done a variety of different things with surface modification using click chemistry.
Thank you. I'm a student at Hacettepe University, Faculty of Medicine. As far as I know, for Alzheimer's disease, there's a cascade that starts with the amyloid accumulation, then neurofibril formation. My question is, as a medical student, to cure my patients, I always consider to treat the initial factor of the disease. That's the amyloid accumulation. So my question is, why are you working with the neurofibrils, not the amyloid accumulation?
Yeah, so the amyloid accumulation is part of it is the formation of the plaque, I guess. And if we can break down this formation of the plaque, or we can inhibit it, I think that is a good case. You have the amyloid not only by the Alzheimer peptide, but also of the alpha-synuclein. And we are not working on that. I guess that you have similar processes with that. And I guess that is actually happening even in the healthy brain. Both the synucleins and the alpha-synuclein and the Alzheimer peptides are present also in the healthy brain. They're just a ways of getting rid of it before it becomes a problem. And the toxic part of it is, as well known, not the fibril itself. Once a fibril is established and it's very insoluble, that's not a real problem. The real problem is these oligomers, the donuts that I showed, and so on. They are the toxic species. And those are probably the ones that forms in small amounts. Once they start forming smaller fragments of the Alzheimer, this becomes more soluble. You're forming more self-aggregating material that can have proteolytic activity. And that's what we are actually seeing with 3 to 6 months of lag time for formation of these active, proteolytically active materials. In vitro. In vivo, I don't know what goes on. We are going to look at the transport of these molecules into mice brain containing human amyloids. And then we'll see.
Sesim geliyor, tamam. First of all, thank you for being here, and I'm here.
Go ahead, please.
Thank you for being here and sharing your knowledge with us. No more questions about funding, I guess. My question is about your inspiration. You mentioned that nature has been one of the main sources of inspiration for you. So what advice would you give us for future scientists who are still trying to find their own source of inspiration? Because as an undergraduate student, I struggle a lot to find my path. So I want to ask you about that.
Take a hike into the mountains nearby, yes, and look around and try to understand what is going on.
Go to nature.
Not only by just looking, oh yes, this is a tree, but understand this tree in detail. What is going on with that? Try to, if you have that kind of philosophy behind it, everything becomes a curiosity case. Everything is interesting. And you will find your interest. If you are interested philosophically, you are also interested in terms of the chemistry that you can understand and do. And you can find projects everywhere. There's no limits to the projects that you can find, because we have only scratched the surface. In science, since back in the starting of the 1800s, scientists has always known everything, right? And we still just think we know everything, and we don't. I can guarantee you that. We don't know everything. So there's so much out there to understand. It's a matter of finding out where it is and how to discover it.
Okay, yes, please.
First of all, thank you for sharing your experience and knowledge. My question is, when you fail in experiments, how do you motivate yourself to keep going? Thank you.
Yeah, so that's the hardest question of all questions today. First of all, I sit down and discuss with them very hard discussions on what they did and what they showed. Because even if you have a failed experiment, there's a lot of information in it. So I want them to actually take that interest in understanding what it was that happened. Not only it went wrong, but what actually happened in that experiment. Because in that understanding lies the solution to the problem. So we are always sitting down with all the spectral data and all the things, and then we discuss and find out what is the alternative approach that we can do. And they always seem to be sort of lifted out of the depression when they leave my office, so that's a good thing. The bad thing is that it usually doesn't succeed the next time either, because you have to really look at all the parameters in your experiments in order to get it to work. In particular, if you are choosing a challenging problem, and if you're not choosing a challenging problem, you're not obtaining anything new anyway.
Last question, please.
Hi, sorry. I am chemistry student, and I want to thank for your passion. And I believe in passion always comes the pressure. And my ask, what do you deal with your pressure? Thank you.
Yeah, the pressure has never been in the science, but more between me and the controllers of the funding. So back to funding. So if we can just get a bag of money and no controllers, that would be perfect. And I think that the pressure, the main pressure is actually for me the question we just had. As I said, that's the hardest question, because the real pressure is when I cannot project success to the students and they fail their experiments, then I cannot sleep at night. I simply go around their problem again and again and again, see if I can find a solution, and I really try to inspire them to think in the same way. Don't stop thinking until you have the solution.
Dear guests, thank you all for your insightful questions and contributions. And on behalf of Middle East Technical University, I would like to once again express our sincere gratitude to Professor Meldal for sharing his valuable knowledge and experience with us today. And now we will proceed with the presentation of commemorative gifts and certificates. So I would like to invite METU Vice President, Professor Dr. Tülin Gençöz, to the stage for the presentation.
Yes, thank you very much. Such a pleasure to be here in front of so many young people. As I said in the beginning, this is a real privilege. Just think about it. I'm 72 years old, and you sit there, 25, listening to me. Hmm?
Thank you so much.
Thank you.
Gift from us.
Thank you very much. Thank you.
Thank you again.
Thank you. Thank you for a great
Thank you very much.
session.
Thanks a lot.